In that case, I've got a question. We're starting to look at complex traits. This is the topic for Friday's class. I'm just going to start mixing in a little bit of Friday content today because I don't know what's happening on Friday, nor Wednesday. So every day this week I have to check in and find out for the following day if I'm going to be on jury duty. So tomorrow I'll let you all know if I'm going to be here Wednesday, and Thursday I'll let you know if I should be able to be here on Friday. So I'm going to mix together a little bit of the content. You don't have to have read this for this class. This is on the syllabus for Friday. But human height is an excellent example of a complex trait. What does that mean? What's a complex trait?
More than one gene controls the phenotype.
More than one gene controls the phenotype. Right? If only one gene controlled the trait of human height, how many heights of humans would there be? Well, I suppose it could depend. So let's say there was the height gene, and there was a tall version, HT, allele, that is, at this gene, and there was a short version, H sub S for short. So if this is true, then, yeah, how many heights, total possible heights could there be? It depends. So why would you say?
Well, like if you have codominance or yeah.
Right, so if there's codominance, if you're HT over HT, you'd be tall. And if you've got two versions of the short allele, you're short. And what happens if you're a heterozygote?
If it's dominant, then you're tall, but if it's codominant, you have a new height.
Right. So this is the only place where the assessment differs. Sorry, that's supposed to be an S. Which it doesn't look like now anyway, but HT over HS, the heterozygote, this is the only place where the trait might differ. If the trait's codominant, that is, they might be medium height. You get a blending of the two alleles, essentially. Or if it's just pure dominance, then we'd have to know which allele is dominant, the short allele or the tall allele, and whatever the phenotype of that heterozygote is would tell us which allele was dominant. So let's define the T allele to be dominant. In that case, a heterozygote would be tall. So it's relatively easy to understand how evolution works to select for certain phenotypes when you work on simple traits like this, where there's one gene that controls a trait. Unfortunately, most traits aren't like this, and that's why we have to talk about real populations, as we did last week, and also talk about cases of complex traits. So does anybody have any questions yet? Mostly review from genetics. So simple trait. Within a family, this sort of thing might happen. But what I want to do today is talk, and we're going to do a little bit of small group work and discussion. How does allele frequency change? Things we worked on last week. How does allele frequency change when you have selection on a trait that has multiple genes involved? All of the simulations that we ran last week were just talking about allele frequencies at one gene and how one gene's allele frequencies change over time. If you select for it, it goes to fixation. If you select against it, it goes to drift, or it goes to loss. Neutral alleles vary over time. Eventually, in how many generations will an allele presumably be fixed or lost? There's a relationship that I briefly mentioned last week, that on average you expect any allele in a population to be fixed or lost. It depends entirely on the size of the population.
4 times the population size?
Right, 4 times the population size. So 4 times capital N, a population size. So it's about that period of time you expect to see fixation or loss. Even if the allele is neutral, that is, if the allele doesn't do anything to your fitness, and natural selection doesn't do anything to increase or decrease its frequency, on an amount of time that's 4 times or number of generations that's 4 times the population size, you expect to see any allele to be fixed or lost. So that would be frequency equals 1 or 0. So how does this work with complex traits? That's what we'll talk about a bit today. I'm going to do a little bit of review, again from genetics. This may be familiar, it might not be. When we have a simple trait, like we talked about at the start of class, if height were simple and there were only 2 alleles, this would happen. So if we were talking about pigmentation of mouse fur, for example, you might have a situation where you have a little a allele, we're talking about one gene. So if you're a heterozygote, little a over little a, add the slash in between them, then you have no copies of an allele that produces pigment in the fur. So this individual, the color of the square indicates the phenotype. So the circles indicate the genotype, 2 white, very light little a alleles, and those 2 little circles produce a light coat color. And then we imagine a case where we cross it to a parent that's got dark brown coat color, and that's because that individual has 2 alleles that are the black circles, both of which deposit some pigment into the fur. In this case, these appear to be codominant because in the F1 generation you get mice with intermediate coat color. So we can say that if you have 1 black allele and 1 white allele, you make half as much pigment as if you had 2 black alleles. Everybody okay with this? So it's kind of like it's additive. It's a diploid individual. They've got 2 alleles of this gene. If they have 1 capital A, they produce some amount of pigment, and that makes their coat color at least intermediate. If they have 2 capital A alleles, they produce twice as much pigment. Their coat is darker. And then if you happened to have 2 heterozygote individuals mate, they produce the usual 1 to 2 to 1 phenotypic ratio, Punnett square, all of that. And so you can see that even within siblings, if you have 2 little a's, you get a light coat color. Heterozygotes have an intermediate coat color, and the homozygotes for capital A have the dark coat color again. So we have variation in the phenotype. So that's how you tell a simple trait. You get a binary or trinary, depending on whether it's simple dominance or codominance. So under codominance, which is like this example, you have 3 traits, or 3 phenotypes. You'd have the recessive phenotype, the dominant phenotype, and the heterozygote phenotype. Or, under simple dominance, you just have 2 phenotypes. You have the dominant phenotype and you have the recessive phenotype. So that would be how you would identify whether a trait is simple or not. So in codominance, you see a 1 to 2 to 1 ratio of phenotypes. What would the ratio be if there was simple dominance happening? The phenotype ratio for simple dominance would be 3 to 1. Those 3 would be, in the case of simple dominance, the homozygote and the 2 heterozygotes. And the 1 would be the homozygous recessive genotype. So let's say we had a bunch of individuals, something an evolutionary geneticist would want to do. We see variation in a phenotype. So we've got a number of individuals here. I'll just number them. So we've got 8 individuals. We've got their genotypes. So you can imagine here I'm circling on the left the genotype at 5 different genes, B, C, D, E, and F all along one chromosome. They don't have to be on the same chromosome. So we've got the genotype of individual 1, and we've got its phenotype here down below. So somehow that genotype, capital B over capital B, capital F over capital F, etc., produces this phenotype. And we want to know what gene controls this trait. So I've shown you 8 different phenotypes, 8 different genotypes. This genetic mapping of a simple trait. How do we figure out which of the genes controls this trait of skin color or coat color?
Which one is different from another?
So we want to know, we're looking for, carry on with that thought. How do we figure out which of these genes controls these traits? It sounded like you were going in the right direction.
I'd probably start looking at one that, so like, that seems dark to be.
Yep.
Look for something that is, the same, like 2 capital letters.
Okay, so you could start with phenotype 1, and you could say the assumption, just pick an assumption.
Yeah.
Assume that
So
there's a
I'm
dominance.
assuming that like B, capital letter, the first one, is what's producing that. And so then I'll go to the second one and see if that could produce whatever is below it and then go down the line.
Okay, so what I'm hearing is there's a hypothesis that a capital letter, this is dominant. That's fine. So you start with the assumption that there's a dominant allele that produces pigment. So capital B over capital B is a good place to start. So then you said you go down the line and you ask, is that hypothesis that dominant capital B allele produces pigment? So what happens when you go to individual 2 at this gene? So it's heterozygous, and the pigmentation is a little bit lighter in this individual, down here in the bottom. So that would be consistent with a codominant trait. If you get 1 capital B, 1 little b, you've got some sort of intermediate trait, which would predict what? What should happen when you go look at an individual like this that has the lightest coat color?
Something else.
Yeah, you'd predict that it might have, for example, 2 little b's, if this is recessive, but it doesn't.
Can you start with looking at the phenotype? Like, you have 2 dark brown phenotypes, why don't we look at 1 and 4 and see what's the same and see what's different?
Okay, so let's look at phenotypes 1 and 4, the 2 that are the same, and find out what's common about their genotypes. It's an equally reasonable way to go. Both are fine. So what's
2 big F's.
consistent? So we've got 2 big F's. So both of those individuals are homozygous for capital F. Now, presumably, the hypothesis is still we're talking about dominance.
So if it's codominant, then it would be an intermediate with those other colors, and it would be a big F and a little f. And then if it was
Right.
homozygous, recessive, then it would be the lighter color, 2 little f's.
So if you take all of the individuals that have the intermediate coat color and you look at their genotypes at this gene, they're all heterozygotes. And if you look at the lightest phenotypes, and you look at their genotypes at gene F, they're both homozygotes. So this is a great hypothesis. So far, the hypothesis that you've generated is that capital F is dominant, controls pigment production. If you've got 2, your coat is darkest. If you're heterozygote, you're an intermediate. And if you have 2 little f's, you have a light coat color. And so far at gene F, this is completely consistent with that hypothesis. Now, that's not to say that other hypotheses couldn't possibly be true. But this is so all we do when we look for what gene controls a trait is we're looking for this correlation. And the stronger the correlation between phenotype and genotype, the more likely it is that this gene does actually control this trait. So I might conclude, as it seems you did, that gene F likely controls this trait, and not B, D, C, or E, unless somebody has come up with a hypothesis for how one of those genes would control this trait. So that's an example of mapping a simple trait. You look at one gene, you look at the correlation with the phenotype, and you just look for consistency. Do all the individuals with one genotype have one phenotype, or vice versa? Now, how do we do this for complex traits? Unfortunately, there are not many simple traits that are interesting to geneticists, evolutionary biologists to study. We love studying simple traits because they're simple. Less work for us. But a lot of traits that people are interested in evolution aren't just controlled by a single gene. So what do we know about how many genes control human height?
16 or something.
I don't know, but it could be 16, sure. We'll go with 16. Beyond that, what else is true about human height?
Environment has to play a role, right? Is that accurate?
Environment probably plays a role as well. So it's pretty difficult for geneticists to figure out by looking at so if we had the genotypes of all of these individuals, they've been lined up here by how tall they are in 1-inch increments. And we can see that there's variation, this discontinuous variation, or continuous variation rather. It's not discrete, it's not binary, it's not light, medium, dark, coat color, it's not short, medium, tall. If it were, that would suggest that this is controlled by a single gene. If there were discrete phenotypes, if everybody was either 5 foot or 5'5" or 5'10" or 6'4", but we see continuous variation in human height, that tells us that this trait is caused either by lots of genes working together, or the environment, or both. So then how are we supposed to know if height is something that natural selection can act on? Does it have a genetic basis? Is height selected for in humans, do you think?
Some people.
Some people. Basketball players? No, see. Volleyball players. Only if the basketball and volleyball players mate with each other, I suppose. So there is genetic variation in human height. It's just a lot of genes control this trait. So complex trait, you will see like this, continuous variation, not discrete phenotypes. See individuals of every height. And that's what this histogram in the bottom right is meant to represent. So instead of a simple trait, you'll either see, as we already discussed, 2 phenotypes, or 3, depending, 2 if it's simple dominance between the alleles that you have, 3 if there's codominance, or incomplete dominance. In a complex trait, you'll see a variety of phenotypes. So one textbook uses this example, again dealing with coat color, where instead of 2 genes, we have 3 genes, A, B, and C. So now instead of this previous example, there are 3 genes, 6 possible alleles. So every time you get a capital letter, you get a slightly darker coat color. So instead of A's and B's, now we have A's, B's, and C's. There's 6 different levels of coat color. You have no black alleles, you've got 1 black allele, 2 black, 3 black, 4 black, 5 black, 6 black. So when you've got 6 black alleles, that gives you the darkest coat color, 0 out of 6, the lightest. How do we figure out the genes that control this trait? So here's our example for this one. We have a plant that has leaves that have lots of hairs and a plant that has leaves with very few hairs on them. So we call parent 1 the less hairy plant, parent 2 the more hairy plant. And we have their genotypes on the chromosomes that are shown here. We've got plant 1 has red chromosomes, the red versions of the alleles, and parent 2 has the blue chromosomes. Is this dominant or codominant? Or incomplete dominant? What happens in their F1? Does it look like either parent? It's kind of intermediately hairy, so it's not pure dominance, it's not simple dominance. So if you're a heterozygote, F1, you've got one red and one blue set of chromosomes, you have an intermediate phenotype. So that's incomplete dominance. So we have to do exactly the same thing. This time I'm not showing you letters to represent genotypes. This time we just have these images that represent genotypes. So all along one chromosome from top to bottom, this individual has all of the red versions of the alleles. And by the way, although they're only drawing one chromosome in each of these F8 individuals, this really means that this individual's diploid. They've got the same genotype on both chromosomes. So that individual next to it looks like that in terms of genotype. It's diploid, but it's homozygous at every locus. So if every one of those individuals is homozygous for every gene, do we expect to see any partially hairy or half hairy leaves like the F1? So the idea is that if you're heterozygous at the gene that causes the trait of hairiness, then you're going to have that incomplete dominant or sort of blended phenotype. It's kind of like an intermediate between the two P0 parents. So we don't see any down here in the F8 generation, we don't see any plants that look like the F1. They're either like parent 1 with very few hairs on the leaves, or they're like parent 2 with lots of hairs on the leaves. So with these genotypes and these phenotypes, we should be able to do exactly what we did earlier with coat color. We should be able to figure out which gene controls this trait. So we just have to look for that correlation. So where's the region where the genotype on the chromosome agrees with the phenotype? And remember, parent 1, the red was less hairy, so we should be looking for regions where the less hairy plants have red genes. And at the same locus, we would expect the hairier plants to have blue genes, the blue alleles of those genes. Is there any spot along the chromosome from top to bottom where that's true? If we number these individuals, which two individuals can you look at that immediately dissuades you from thinking that this is possible?
Number 3.
2 and 3.
Okay, so 2 and 3 together. Those individuals have really hairy leaves. They look like parent 2. Should have both blue alleles, right? But there's no place along this pair of chromosomes where both of those individuals have blue, right? Where this individual has blue genes, this one has red. This one has blue genes, that one's got red. I should have worn my blue jeans today. This one has blue, so individual 3 has blue genotype down here, but individual 2 does not. So there's no, even between those two individuals, 2 and 3, that have totally different genotypes, they have the same phenotype. And then you can look at individuals 5, 6, 7, and 9, and further convince yourself that all 6 of those individuals that have very hairy leaves don't really share one particular genotype. It's disturbing. But this is not surprising when we're talking about a complex trait. So this is supposedly an example of a complex trait. I'm asking you to tell me what genes, what combination of genes produces these phenotypes. So we've already, just by looking at two individuals, individuals 2 and 3, and seeing that they share a phenotype, but they don't share a genotype, suggests that this is not a simple trait, not just controlled by one gene. It's probably controlled by multiple genes. And so I'm just going to briefly walk through the type of analysis. I'm not going to actually ask you to do these sorts of analyses, but I just want you to be familiar with how this process works. When you're trying to figure out how multiple genes might control a single trait, like the hairiness of leaves on a plant, you put the genotype information, like the colors of the chromosomes, and you put the phenotype information, whether or not the leaves are hairy, into a particular computer program that does this for you. And what it does is it looks at every, so you start simple. You say, tell the computer, I assume this is a trait controlled by 2 genes. And so what you ask the computer to do is to look at gene 1 and gene 2 and ask, if you look at both of those together, the genotype at gene 1 and the genotype at gene 2, and you look at every individual, does that combination of genes correlate with the trait? So in this case, you could start with genes 1 and 2 and say, well, if this trait of hairiness of leaves is controlled by 2 genes, and both of those genes have to be red, the red allele, to produce a less hairy leaf, then this individual's genotype matches that phenotype. But what about individual 2? If you look at the combination of gene 1 and gene 2, it's not red at both of those loci, so you wouldn't predict that its phenotype would be the blue or very hairy genotype. So then you say, okay, computer program says, okay, that didn't work, so forget about the combination of genes 1 and 2. What about genes 1 and 3? And so the computer program looks at the genotype of every individual at that combination of genes, 1 and 3, and compares that to the phenotype. And then it does 1 and 4 and 1 and 5 and 1 and 6 and 1 and 7. So basically the computer program, because you don't want to do this by hand, takes every possible pairwise combination of genes and asks, is that combination of genotype explaining the phenotype? And if it's not 1 and 2, 1 and 3, 1 through the end of the chromosome, then it goes 2, 3, 2 and 4, 2 and 5. If you want to tell the computer program that it must not be 2 genes, it must be controlled by 3 genes, then the computer program looks at every possible combination of 3 genotypes. And it does this over and over again until you find that set of multiple genes, when you consider them all together, perfectly correlates with the trait that you're looking at. Blah, blah, blah. So that's the approach. In this case, this is the sort of answer that a computer program would come up with. So it looks through all pairwise comparisons of genes. And this is the answer that gets spit out by the computer program. If you look at these 2 genes, which it's calling now gene 1 and gene 2, or locus 1 and locus 2, does this make sense? Does the genotype, when you look at both of those genotypes together, correlate with the trait? So for the individuals that are not hairy, what are their genotypes? When you look at the combination of gene 1 and gene 2 together. They're all red and red. So it's like, to get less hairy leaves, not only do you have to have a particular genotype at one gene, you also have to have a specific genotype at another gene. That's why it's complex. Multiple genes contributing to this trait. Now, what's true in this hypothesis about the genetic basis of this trait, what's true about the hairy leaves? How could you describe their genotypes? We've got 2 genes. Pardon? Would you say that blue is dominant? So, yeah, every individual that's got a hairy leaf has at least one blue allele. at least one blue gene. So that suggests that you have to have two blue alleles or one blue allele at one gene to get hairy leaves. So that does tell you something about the dominance of the trait. But in this case, this is the best correlation we can get between the genotype and all of the individuals and their phenotypes. So it seems like the hairiness of leaves is controlled by at least two genes, both of which have to be red for the trait of being less hairy. That's all the genetics for today. Now the question is, how does this impact evolution? We've been talking for the last week or so about allele frequencies. And what happens to allele frequencies over time if those mutations are neutral, if they don't affect the phenotype, or if they're beneficial. So the question basically is, when a trait is complex instead of simple, how does that affect how it evolves, or how it's maintained in the population after it evolves? So let's say you've got a really important trait to the health of an individual, but it's a complex trait. You've got a trait that's super important, but you have to have the right combination of genotypes of lots of genes to get that trait. It's easier to mess up. So if you've got that trait, what's the likelihood that your kids have that trait? Let's say it's a trait controlled by 5 genes, and you have to have a specific genotype of each of those 5 genes to get the most beneficial version of the trait. It's going to be pretty hard to ensure that your kids get that set of genotypes as well. So I've got a little smaller group exercise for you. Like, please, everybody participate. We're going to discuss this by the end of class. We've got about 15 minutes or so to discuss. And then we'll come back together. Maybe 10 minutes to start. So just like this plant example, imagine that you've got a trait that depends on 2 loci. We call them gene A and gene B. And just like that plant example, there are 2 alleles. So we'll call them the red allele and the blue allele. Allele 1 and allele 2. So in either of these 2 situations, with either of those 2 genotypes, you get healthy offspring. So you either have to have the red allele, homozygosity for the red alleles at both genes to be healthy, or you have to be blue and blue at the other, at 2 loci to be healthy. If you don't, then your fitness suffers. You have less kids. And then imagine that you cross 2 individuals that have the same dihybrid genotype. Blue over red at gene 1 and blue over red at gene 2. So I'd like you, in small groups, I'll come around and answer questions if you have some. Try to discuss and come up with your responses, or not necessarily correct responses, but your responses to these 3 questions. After you go through 4 times whatever the population size is, it doesn't matter what the population size is. After 4 n generations, what would you expect if you went back to this population that was established by a dihybrid cross? What would the genotypes of all of the individuals in the population be? Second question is related to the first. Which genotypes do you not expect to see after that many generations go by? And then the third question, after, again, that same number of generations, when you look in the population, what's the allele frequency of A1, A2, B1, and B2? You've got all the tools that we talked from the first half hour. You've got the tools to answer this question, this set of questions. So work together and see if you can come up with some answers to these. We'll shoot for about 8:43 or so, coming back together. 10 minutes. Okay, so what sort of genotypes do you expect to see after basically a very large number of generations go by? Selection happens, allele frequencies change or don't. What are the genotypes you're going to expect to see in the population when selection is actively acting on these genotypes?
There's so many.
Which one?
There's so many.
There are so many genotypes, right? There are 16, that's why I drew the Punnett square up there. There's 16 different genotypes, different combinations of these 4 alleles. A1, A2, B1, and B2. Which are the most healthy? So I already told you which ones were most healthy, right? A1, A1, B1, B1, and A2, A2, B2, B2. That is, those really rare individuals, 1 out of 16 homozygous red and 1 out of 16 homozygous blue. So 1/8 of the population in the first generation, after you set up that initial dihybrid cross, 1/8 of the population has these genotypes that are presumably the best fit. But that's just after generation 1. That's the F1 generation. There are all these other genotypes. So what happens to all these other genotypes over who knows how many generations, hundreds, thousands, millions, depends on the population size. Would you say those are the only 2 genotypes you expect to see? Why or why not?
I would say yes, because that they're the most fit, so that they'll produce the most healthy offspring.
Okay, so if they're the most fit and they produce the most healthy offspring, then you might see over an infinite number of generations that those are the only genotypes that are left, maybe. That's one hypothesis. Others? Yeah.
What about the 2 middle ones that kind of complete the diagonal there? Because they're both homozygotes,
Right.
but like, is it necessarily prerequisites that it has to be the main color ones?
Yeah, what I didn't say in this example, which thank you for pointing this out, I meant to say that I was hoping that we were focusing on the paired colors. But yes, you're right. So the way I wrote the question, any of those on the diagonal would be healthy. I intended it to be these that have the matching colors that are the most healthy. So you might say that those are the genotypes you'd expect to see after 4 times the population size number of generations. Go ahead.
So like, I was confused when we were talking about it, like, could you really see both and have just them? Because they're gonna reproduce and produce less healthy offspring.
Well, and that's why having these so if you have a homozygote like this, if they mate with other individuals of the same genotype, then they produce only individuals with that genotype. So you're absolutely right. If there is assortative mating, if the red-only individuals mate only with other red-only individuals, and blue-only individuals do the same, there's assortative mating based on genotype, then you would only have those 2 genotypes in the population. But if they're randomly mating, which we should expect, then you would see all of the possible genotypes showing up. So that's a great point to make. Depends on how mating is happening. But it's sufficient for me if you said you expect to see many of the other less fit genotypes, all of the ones that aren't circled in black there in the upper right-hand corner, to be less represented after 4 times the population size number of generations. Right, you expect the fittest genotypes to be prevalent in the population and less fit genotypes not so much. But yes, we have to recognize that every generation mating is going on and new genotypes that might be selected against are still being reintroduced back into the population. But this is the critical point, and this is setting up for class next time. Question 3. After this many generations passes, What's the frequency of A1 and A2 in the population? That is the red version or the blue version of gene A, 1 or 2, better than the other?
If they both equally fit, then no.
Right. So if the all blue individuals and the all red individuals are equally fit, then you would expect potentially to see as many A1s as A2s, or red alleles and blue alleles. So in sum, what happens is after that many generations, you might see only those individuals and those individuals, you might, in the population after a large number of generations, because those are the fittest genotypes. But would you have detected a change in allele frequency? What was the allele frequency we started in the initial cross in the F1 generation?
A1, A2.
Right there. So what's the frequency of A1 and A2? Yeah, they're both at 50%. Each of those A alleles, version 1 and version 2, are at 50% frequencies. And the same is true for B1 and B2. They both represent 50% of the population. And after an infinite number of generations, the allele frequencies don't change in this example. So you go back to the population after hundreds or thousands of generations, you sample a bunch of individuals, and they all still have 50% of the alleles in the total population are still allele 1, 50% are allele 2. It's just that you no longer see heterozygotes. So that's what's changed, is not the allele frequency over time, but the frequency of genotypes. You could start with all 16 of these possible genotypes, but after selection works, generation after generation after generation, you get rid of those heterozygote genotypes and basically wind up getting a population that's got 2 genotypes. So next class, hopefully, if I'm here, we'll continue this conversation about population genetics. We'll be talking about Hardy-Weinberg equilibrium, which this is an example of. And what I would like to do, please, between now and 11 is answer a couple non-scientific questions. I'm going to launch a Socrative quiz. Basically, I'm just asking for your feedback about how the last few days went. I want to know some things about that, so in case we go on strike, I know how you preferred to handle a class when I'm absent. It's an opinion poll, not science. But please do that in the next 2 hours. I'm going to use that for attendance for today. Thanks.